evidence is a crucial part of crime scene investigation. To start, make sure you always put on your gloves to prevent your DNA or fingerprints from contaminating the crime scene. There are many ways DNA evidence can be collected. One is through the collection of hair follicles. Another way is through the collection of blood samples. Forensic scientists use swabs to collect even the smallest amounts of blood. The evidence is then taken to the lab for analysis and profiling.
Do not need you to check up on me, so please remove yourself from my laboratory!
Step 1 is to extract DNA from the evidence found. Step 2 is to cut DNA using restriction enzymes to better recognize the unique patterns in DNA bases. Step 3 is amplification. Specific DNA fragments are amplified using a polymerase chain reaction. The purpose of this is to amplify and multiply DNA fragments for better analyzation. The final step is electrophoresis. DNA is inserted into wells found in agarose gel. An electric current is charged through the gel, which causes the DNA fragments to move. Negatively charged fragments migrate to the positive end of the gel. The fragments are then separated by size for further analysis. Often, there are so many DNA fragments that they are nearly impossible to analyze. Using radioactive probes, we can identify specific DNA sequences. Each individual person has a unique DNA profile. When DNA evidence is collected from a crime scene, it is used to create this DNA profile. This information can be compared to the known DNA profiles in the databases of suspects. These profiles are entered into CODIS to identify the possible suspects. Using CODIS, DNA profiles can be compared to determine if specimens come from related or unrelated individuals. Every DNA chromosome contains genes. An allele is one or more alternative forms of a gene. These different forms are what give a person their unique qualities like hair and eye color, and are the reason no person is exactly the same as another. Every person gets one set of alleles from your mother and one from your father. This is why half of your DNA is from your mother and half of your DNA makeup is from your father. Using DNA profiles, you can compare this information to determine whether or not a person is directly related to another.
Alright, V and TR are variable number of tandem repeats. These are short sequences of DNA that are repeated multiple times. For example, CATAC, AGAC, CAT, ACAG, AC.
As you can see, it is CAT, CAT, and these are repeated strands of DNA.
The human genome is full of repeated DNA sequences. These repeated sequences come in various sizes and are classified according to the length of the core repeated units, the number of units, and the overall length of the repeat region. DNA regions with short repeat units are called short tandem repeats, or STRs. STRs are found surrounding the chromosomal centrum. STRs have become a popular use in forensic science. STRs have become popular because their DNA markers are easily amplified by polymerase chain reactions, without the problem of differential amplification, that is. The PSR products for the STRs are generally similar in amount, making analyzation easier. An individual inherits one copy of an STR from each parent, which may or may not have similar repeat sizes. The number of repeats in an STR markers can be highly variable among individuals.
which makes these STRs effective for human identification purposes. Mr. Simpson, even though the DNA evidence has shown that the blood on this glove belongs to your ex-wife, does it fit? It does not! Well then, case closed. And of the core, Mr. Simpson, even though the DNA evidence has shown.
You're doing so good.
It's okay.
Now, here comes the music. Boom, 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 boom. I'll shoot you right down. Boom, 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 boom. Ow, 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 ow. Boom, 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 boom. I'll shoot you right down.